So probably many of you aren't familiar with me or my work. We're relatively new to UF. We just joined in July. So I want to give you a little bit of an introduction into some of the kinds of work that we do, a little overview of our lab. So as was mentioned, we do a lot of work in perception. So we focus on language perception, music perception, print perception, and reading, with a focus on reading network development in both typical readers and those with dyslexia. And we also do some work with non-invasive vagus nerve stimulation. I'm actually not going to talk about any of that work today, but I'm happy to later or off to the side if anyone is specifically interested in the vagus nerve stuff. And to answer the questions that we have in the lab, we use a variety of tools, lots of neural imaging, EEG. We're excited to get back into using fMRI now that we're here at UF. As I mentioned, the vagus nerve stimulation stuff. And we do genetic testing in humans. We also do some genetic manipulation work in rat models. So being able to really dissect the individual contributions of specific genes to reading and language, and then testing those predictions in humans. So today, we're going to focus on dyslexia. Again, happy to talk about some of the other work that we do in other areas if anyone would like. So reading is a very critical skill for daily life. It's something we do every part of our personal lives, as part of our academic lives, as part of our vocational lives. And reading is something that we expect all kids to learn and to do well and automatically. Even though reading is required, reading is not an automatic skill for the brain. So we think about language as being this hardwired skill. So a baby is born, the circuitry is there and ready to support language. If you put a baby in a room, as long as there's exposure to language, the brain will kind of figure it out, right? But reading is not that way. If you put a kid in a room full of books, the kid just doesn't learn how to read by osmosis. It requires explicit instruction. Reading is a human invention, dates a few thousand years ago. I think 8,000, 8 to 10,000 is a current estimate of when reading was initially invented. And so because of this fairly recent invention, the brain hasn't had enough time to develop an automatic network for reading at birth, which means that in every kid learning to read for the first time, the brain has to figure out, how do I support this skill that turns out to be incredibly important for my daily life? So there's a region of the brain on the fusiform gyrus, it's the bottom surface of the brain, that we call the visual word form area, or the VWFA. The VWFA is a region of the visual cortex that becomes responsive to print over the course of reading instruction. So by the time a kid is a fluent reader, we have this beautiful region of the brain that responds preferentially to print over other kinds of stimuli. Of course, this doesn't happen magically. The VWFA requires connections to other parts of the brain in order to develop that expertise. So over here on the right-hand side, you see that area in red showing you the location of the visual word form area. And you'll note that it has connections to these canonical language regions that we think about, responsible for phonological awareness, speech sound perception, speech production areas in the front. So this region of the brain was first described around the early 2000s, and it requires experience. It requires training in order to develop. Prior to a kid going into school and learning how to read, this region of the brain you see here kind of on the left side of this cartoon schematic, this part of the brain is doing other things, right? We're not born for readers. So this part of the visual cortex is doing things like processing tools and objects and faces and other really important kinds of visual stimuli. If a kid goes on to go to school and learn how to read, those neurons start to change their job. They start to become print processors. And so you see that over there in the top right, those areas in green, these parts of the brain that become responsive to print and words in kids that learn how to read. Kids that don't learn how to read don't develop a VWFA, right? So if you are not taught to read, if reading doesn't become an important skill for you, then those neurons just keep doing what they're doing. They do more tool processing, they do more face processing. And what's important too about the VWFA is that it maintains a very long plastic life. So the VWFA can change a lot over the course of the lifetime. Just because you didn't learn to read in childhood doesn't mean you can't, doesn't mean this area will never develop. In fact, if we look at adults who were previously illiterate, as individuals learn how to read, the VWFA comes online, even in adulthood. So in this figure, what you see in the middle of those three panels, that dot there in purple, I'm not sure if my mouse will work, but I will try. Let's see. So this dot here in purple, these are illiterate adults that don't read. So of course, on the x-axis, you're seeing they're not able to read any words at all. And on the y-axis, there's very little activity in that visual word form area. But there are these two dots here in blue. These are adults who used to be illiterate and then learned how to read in adulthood. And so you see they have some more visual word form activation. And importantly, there's a relationship between how strongly the VWFA responds to print and how well you read. So the stronger your print response in the brain, the better a reader you tend to be. This visual word form area is hypoactivated in dyslexia. So activation to print in this area in individuals who struggle to learn to read. And it turns out that that is present very early on in life. So this is data from a longitudinal study we did a number of years ago now, looking at kids who were at risk for dyslexia. So we brought them in around the age 4 or 5. We did an fMRI scan, and then we followed them several years in the future. And the goal was to understand whether this visual word form area hypoactivation is present from the beginning, or if it's a consequence of poor reading or reduced experience. So within our group of at-risk kids, about half of them went on to become typical readers. It's a pretty standard proportion of kids who are at risk. Half went on to be typical readers. So that's what you see in that middle shade of gray. And then about half went on to have a dyslexia diagnosis. And what was really interesting in this sample is a pretty clear division in the brain activity. So we presented letters, you see over here on the left, and then false fonts. So these are images that have all of the visual characteristics of letters, but they have no meaning, right? So this object over here on the right has all the same curves, all the same lines as the letter B. They're just kind of shuffled around, right? So in our kiddos that went on to a dyslexia diagnosis, you'll notice that the visual word form area responds significantly less to letters and false fonts compared to their peers who were either not at risk or kids who were at risk but went on to develop typical reading. Now, even though this is a very common neural deficit in dyslexia, it's the most well-replicated neural deficit in dyslexia, we know that not all kids with dyslexia behave the same. They don't all look the same. And we've known this for a really long time. You go back into the 70s and you'll find papers like this, a sample of kids with dyslexia. Some of them have visual deficits, spelling deficits, others have auditory deficits, some have combinations. We know there are different behavioral profiles within the population of kids with dyslexia. Over time, it's changed a little bit how we talk about it, how we think about it. Double deficit hypothesis is probably still one of the most prevalent hypotheses in dyslexia, this idea that kids with dyslexia have either difficulties with phonological awareness, understanding the building blocks of speech, the small little speech sounds that make up words, or deficits in rapid automatized naming, or RAN, this ability to look at a letter and just automatically know the sound that it makes. You don't have to sound out every single letter, very automatic skill, or deficits in both. So this is kind of the framework that we tend to think about even now, but we still don't have a great handle on how do we use behavioral data to help these kids. So we can't really predict what their profile is going to look like by the deficits they have. So this is a relatively recent-ish study, basically looking at two different deficits that are reported in dyslexia. So you have phonological awareness on the y-axis, and this is visual attention span on the x-axis. And what they found was that even though groups of kids that differ on these two skills, even though they seem to have a little bit of a behavioral difference in decoding, so reading non-words, the behavior alone was not enough to differentiate their profiles. So we still have a lot to learn about what is causing this heterogeneity, but we know that it's there. Current interventions in schools, I think, attempt to deal with this. There's a number of different interventions currently out there. There's a handful here on the slide. Typically, this is pull-out instruction in schools by SLPs or reading specialists, or one-on-one instruction in a clinic. It's typically how these things work. And these interventions are really a combination of skills. So you're introducing new concepts, or you're practicing targeted skills. So things like phonological awareness, word reading, sentence reading, spelling, comprehension. So it's generally an umbrella approach. You're targeting lots of different skills. But even though we're covering a lot of information, this approach is not tailored to individual kids. So kids are going to come in, they're going to get this intervention no matter what their individual phenotype profile is. And so maybe it's not surprising that a lot of kids are not responding to the interventions that we have. So there was a study that came out just last year. They followed 141 kids that were involved in the Take Flight intervention program. Of those 141 kids, only 80 of those kids achieved what they called an adequate response, which was meeting two different benchmarks that they had. So a reading benchmark and a curriculum benchmark. So 80 out of 141 kids are responding to this intervention. About 26 of those kids met one of the benchmarks, and 35 met neither benchmark. So there's something missing here. We're missing, how do we target kids that maybe don't perfectly fit into the buckets that we generally consider. So our lab takes a biological approach to understanding heterogeneity, and we feel like understanding the biological mechanisms of dyslexia may provide some additional insight to help us improve intervention for these kids. So we're going to do a very, very brief genetics primer. This is not a deep dive into genetics talk, but we do focus on genes that are dyslexia. We know there's no single causal gene associated with dyslexia. So there's a number of genes that are associated with dyslexia, and not any one of them on their own cause it. And so a handful of these are genes that we study in the lab. These are two that I'm going to focus on today. They're neural migration genes. They live on chromosome 6, KIA0319, and DCDC2. So neural migration genes basically are involved with brain development in utero. So making sure that neurons get to where they're supposed to be, they go to the right neighborhoods, they become the right kind of neuron. And so these two genes are commonly associated with dyslexia. So since there's no single gene for dyslexia, a lot of the work that we do is focused on understanding what each gene does to the broader phenotype. So what is KIA0319 doing to cause elements of dyslexia? What is DCDC2 doing to cause elements of dyslexia? There we go. So in humans, KIA0319 is associated with reduced activation in the left temporal lobe. So you see that area highlighted in yellowish orange color here. We've also found that it's associated with poor neural consistency in rat models. I'm going to tell you all about that in just a minute. DCDC2, on the other hand, is associated with a different profile. So in DCDC2, we see this is linked with reduced white matter tracks in the reading network. So think about white matter like information highways, if you're not familiar. These are connections between brain areas. So if you have reduced integrity, think about a road full of potholes, right? It's harder to travel that road if the road has less integrity, less structure. We've also seen this gene linked with perceptual speed challenges in animal models. And again, I'm going to tell you a little bit about that in just a bit. So let's start with KIA0319. If I say the word dad several times, your brain is going to respond generally the same way every time. And that makes sense, right? You hear a word, you want to make sure that every time you hear the word dad, your brain knows what you just heard. So here I've just got a little cartoon schematic of 3 times that we've presented the word dad. The black vertical line indicates the onset of the word dad. And then those little vertical kind of bluish green lines might represent the onset of the neural response. So generally, the brain is pretty consistent, plus or minus about 4 milliseconds. From the time we present the sound dad, your brain's going to respond consistently the same way every time. But in rat models, when we manipulate the gene KIA 0319, what we found is that the brain was a lot less consistent. So we basically turned down the expression of this gene in the auditory cortex in rats. And in those animals, what we see is this increased variability. Sometimes the brain responds too early. Sometimes it responds too late. Sometimes it responds just perfectly on time. So if we're thinking about speech sound perception, if the auditory representation of a speech sound is not consistent, if every time you hear a D, it sounds a little different to you, then that would make it really hard to match that letter to the sound that it makes, right? If the sound's not consistent, how are you going to know what sound goes with what letter? Of course, this is all done in animal models. Rats don't read, right? So does this mean anything in the context of dyslexia? So we ran the study to find out. So this was a study done in magnetoencephalography, or MEG. This is a wonderful technique. It is not loud, it's not claustrophobic, it's really comfortable. Kids are perfectly fine to come sit in this comfy chair and do some tasks with us. And so we brought kids into the lab. They sat in this big comfy chair for about 20 minutes, and they listened to a bunch of sounds, tones, and speech sounds, and they looked at a bunch of visual stimuli, letters, and some nonsense objects. These kids are about second graders, so about 7-ish to 12 years old. We brought them in, we screened out for a bunch of things, IQ, language, reading. I'm happy to talk about the individual measures that we used if that's of interest, but for time, I'm just going to kind of skip through and tell you that we verified that all of the kids with dyslexia had dyslexia and nothing else, and our typical readers were in fact typical readers. So we looked at how the brain responded across all those different repeats of the different sounds. And so we pulled out activation from auditory cortex, that's the area you see in black on the brain image, and we calculated variance. So how variable is the brain's response across repeats of the same sound? So pretty obvious group result, group difference here. We found our kids with dyslexia had significantly higher variability compared to those control kids. So the group of kids with dyslexia, their brains had that inconsistent timing, right, that we saw in the animal model. Now, if we just looked at this, like many people have done for decades, we might say that neural consistency is a causal feature of dyslexia. Look, they all have it. The group difference is very obvious. The bar under the dyslexia column is much higher. So maybe this is the thing. This is a thing that's going on in dyslexia, and maybe they all have it. The problem with that is when you look at group levels, you miss the detail in the groups. You miss the individual variability. So when we looked at the raw data, what we found was this natural split within the data. So about half of those kids, the circles that you see, had really high variability, and the other half looked just like the typical kids. Perfect consistency in the brain, no problem at all. The kids that had really high consistency, our circle kids here, also had variance in that same gene that we manipulated in the rat model, KIA 0319. So that gene, KIA 0319, seems to relate to the consistency with which the brain responds. Kids that have poor consistency in the brain also seem to have variance in this gene, KIA 0319. But of course, it's not the only gene in dyslexia. So what can we learn about other profiles, other subgroups within dyslexia by looking at some other genes? So that brings me back to my other friend, DCDC2. So we talked a little bit about this earlier. I briefly mentioned that DCDC2 is linked with poor white matter integrity, right? So reduced organization, reduced clustering of those white matter tracks. And again, poor perception speed in rodents. So back to the rodent data for a brief second here. This is a summary of the rat data that I told you about earlier. So we have our control animals over on the left in that dark gray, the KIA 0319 knockdowns in light gray. You can see they have a lot more variability. in the response to sound, just like you'd expect. We also manipulated DCDC2 in a separate group of animals. And it turns out that DCDC2 has nothing to do with variability, does not change the way the brain responds to multiple repeats. So the DCDC2 knockdowns in red, they look just like controls. Okay? No issues at all. These animals, though, have a hard time with speed. So behaviorally and on the neural side, when we start making the sounds go faster and faster and faster, all of a sudden their performance crashes. So these guys have consistent brains, but they can't handle speed. It turns out this is also replicated in humans already to some extent. This is work published in 2017 by Nicole Neef and a whole bunch of collaborators looking at a group of kids. Kids that had variants on DCDC2 were more likely to have deficits in reading speed and reading comprehension. No deficits in spelling, which is a letter sound pairing question. Okay. We've also found in kids that this gene is associated with rapid automatized naming, that second deficit that I mentioned earlier in the double deficit hypothesis. So it could be that KIA 0319 is involved with perceptual representation, so understanding the speech sound itself, and maybe DCDC2 is associated with speed. How quickly can you map a letter to a sound? Now, I haven't mentioned in quite a while the visual word form area, but I led with it. So you may be wondering why I haven't talked to you about the VWFA and genes, and that's because we don't know the answer yet. That is still something that we are working on. What gene or genes is associated with that classic hypoactivation in the visual word form area? So the lab is currently addressing this question. We're thinking about this in potentially new ways. We know, again, that kids with dyslexia tend to look different. We have these different behavioral profiles. And so what is causing those behavioral profiles? Is it the case that neural inconsistency and VWFA are hypoactivation are two sides of the same coin? Are they co-occurring? Are they there together? And that leads to a particular phenotype? Or are they separate things? Is neural inconsistency leading to some profile of behavior, whereas VWFA hypoactivation is leading to a separate phenotype of behavior? And are these mediated by genes? We think that KIA 0319 is associated with neural inconsistency. Rat work, human work back up this idea. Is it possible that DCDC2 contributes to VWFA hypoactivation? Remember that the VWFA requires connections to other brain regions in order to specialize. Those connections are white matter connections. So if DCDC2 is impacting white matter and the VWFA requires white matter to develop, could there be a link between DCDC2 and the VWFA? So this is work that we're currently doing. We're gearing up to do in the lab using EEG as well as fMRI to disentangle these two neural features to measure VWFA hypoactivity in the same kids that we're measuring neural inconsistency, in the same kids that we're doing a very thorough behavioral battery to start to try and tie these things together in different groups. Okay. So we could spend hours and hours talking about heterogeneity, but I did want to take a little bit of time just to bring us back to the present. What do we do with all this information as we continue working on it? I think understanding biology is step one. So it's very helpful for us to understand what is going on in the brain, what genes are causing these different neural response patterns, and understanding whether these different whether these different neural features can be influenced by intervention. So our prior work in the rats suggests that the KIA 0319 issue can be fixed by training. Rats that we train behaviorally, their brains become way more consistent. They start to look like control animals. The DCDC2 rats don't seem to be as responsive to intervention. Behavioral training actually seemed to make them worse rather than better. So understanding these biological mechanisms, how they relate to plasticity, might then let us go into meaningful guidance for clinicians and teachers and parents, right? So these interventions that cover lots of different skills, how do clinicians know which skills matter? If there's a kid in front of you who has really good rapid naming, but really bad phonological awareness, it might help that kid to say, well, let's just skip the RAND stuff and let's focus on phonological awareness. Lots of clinicians don't have the training to know how to make those moves in real time. So part of me being excited about being here at UF is all this AI stuff that's going on. We've done a little bit with machine learning in the past, and we're in the process of planning this collaboration to hopefully try and use some AI, some machine learning to help provide some guidance for clinicians on what areas of the intervention might be best to focus on with a particular child. And then the last thing I want to say is this bridging of the gap. So this is not new information to hopefully anybody. I think everybody kind of preaches this bridge the practice, research practice gap. We know so much more in the research labs than what clinicians and teachers know. There's a wave of studies in the SLP journals showing that teachers don't know a lot about the science of reading, how it works. Clinicians don't really know about this genetic neural heritability stuff that we've known now for decades. So there's this gap between the information we have and the people who we rely on to use that information to then go help kids. I'm always amazed when I talk to teachers and clinicians how little of this information they know. And that's not their fault. You know, they are not researchers. They are in the classroom helping kids, in the clinic helping kids. And so a lot of the burden falls on us as researchers to figure out how do we package the information in a way that they can use and easily digest, right? We have lots of hours of experience reading research articles, they don't. So it's a little bit on us to help them understand what we know and to help them figure out how to use it. So I'm not sure how I'm doing on time, but I'm going to go ahead and just stop right there. Thank my labs, all the collaborators, all the funding sources, everything. And I'm happy to take whatever questions anybody has about this or anything else we do.